In March of 1963, Dr. Maurice Hilleman was woken up one night by his 5-year-old daughter. She was complaining of a sore throat, so Hilleman looked her over and determined she had the mumps. Unable to sleep, he was struck with an idea. He swabbed her throat for a sample, drove to the lab, and got to work. 4 years later, his mumps vaccine was approved. It was the fastest a vaccine had ever been developed. Until now.
New vaccine against coronavirus.
And it's incredible how quickly this vaccine and others have been developed.
This was done in record time.
This is extraordinary. This is the
fastest vaccine development in U.S. history.
In 2020, vaccines for COVID-19 have shattered previous records, going from development to approval in a matter of months. That speed was driven by billions of dollars and a global effort. But in some cases, it was also because of a breakthrough in vaccine technology decades in the making. Something that could shrink this timeline going forward and change how we make vaccines altogether. Vaccines teach your immune system how to respond to a threat, and traditionally there have been 4 ways to do this. The 2 most common types of vaccines work by exposing you to a weakened or dead version of a virus or bacteria. The weakened virus won't make you sick, but it will teach your body how to fight the real thing if you're exposed to it later on. This is how the measles and annual flu vaccines work. Another, less common type of vaccine does a similar thing, but uses an inert version of a toxin instead of a virus. The most well-known version of this is probably the tetanus shot. This 4th type of vaccine works a little differently, because it only uses a small part of the virus instead of the whole thing. Common examples of this would be the hepatitis B vaccine or the HPV vaccine. Some of the new COVID-19 vaccines also rely on these traditional methods. For example, one COVID-19 vaccine currently in trials uses this 4th method. It only uses one part of the SARS-CoV-2 virus, known as the spike protein. That spike protein is what allows the coronavirus to enter your cells. When injected into your body on its own, it's harmless. But your body will still recognize it as a foreign threat and launch an immune response to fight it off, which is enough to teach your body how to fight the whole virus. But isolating and preparing that spike protein for a vaccine is a process. Researchers first had to modify it, then multiply it a lot, and then assemble the vaccine itself in a lab. In fact, one thing that all 4 of these types of vaccines have in common is that they all require growing and transporting large amounts of live pathogens in a lab. And that takes a lot of time. A vaccine goes through many steps before it can be approved. But before anything else, it has to be developed. And working with live pathogens makes that process a lot longer. On average, it takes 5 to 10 years for a vaccine to reach FDA approval in the United States. Most COVID-19 vaccines have gotten through this process a lot faster by overlapping the different phases of human trials and starting the manufacturing early. But some vaccines have also found a groundbreaking way to speed up this first section by shifting some of the work out of the lab and into your body. Nearly every function in the human body is carried out by proteins, so our cells are constantly manufacturing them. To do that, they make a single-stranded copy of DNA. That copy is called messenger RNA, or mRNA. Each strand of mRNA holds the information on how to make one type of protein. The cell reads the mRNA, follows the instructions, and makes a protein. And that's where these 2 new types of vaccines come in. They contain instructions. Researchers who developed these new types of vaccines, called mRNA vaccines, started with the genetic sequence of the virus. They also decided to focus on the spike protein we talked about earlier. But instead of assembling and purifying that protein in a lab, they identified the part of the genetic sequence that creates it. And then took a much faster route by synthesizing mRNA and using that as the vaccine, which saved months of time and money. Once inside the body, the cell reads the mRNA and begins to make harmless spike proteins of its own. From there, your body's immune system recognizes the foreign threat and sounds the alarm. This is how the new COVID-19 vaccines from Pfizer and BioNTech and Moderna work. But the main drawback with an mRNA vaccine is that mRNA breaks down very easily. It has to be delivered inside a protective fatty barrier and kept ultra cold, which isn't super ideal for a vaccine that needs to reach all areas of the globe. Another effective new-to-consumer kind of vaccine works similarly, but uses DNA instead of mRNA, which is much more stable. The COVID-19 vaccines from AstraZeneca and Johnson & Johnson are this type. It doesn't require the ultra-cold conditions, but it does have its own drawbacks. To get the DNA into your cells, researchers use a harmless virus as a carrier. But over time, your body will build resistance to that virus, which means future doses using this carrier will become less and less effective, and the carrier will need to be updated. But in terms of efficacy, cost, and speed, these two new types of vaccines have broken records. These new vaccines are a groundbreaking way to elicit an immune response. And while they'll have a big impact on how we fight COVID-19, their real impact is just beginning. A vaccine that delivers specific instructions to your body opens up a whole new world of vaccine technologies and disease treatments for things like cancer or HIV. Finding the vaccine was a turning point for the pandemic. But the pandemic might also be a turning point for vaccines.
My name is Stephen Hogue, and I lead our platform efforts at Moderna, together with our platform Chief Scientific Officer, Melissa Moore. At Moderna, we believe messenger RNA represents an unprecedented opportunity to develop medicines for patients, one that could have a dramatic impact on the course of human health and disease. In order to understand why we believe that, we have to pull back and ask a very fundamental question, which is, what is life and how might messenger RNA impact it? Now, life is really just the flow of information in a biological system. The ebbs and flows of that information dictate health and disease. And that's true in every cell in our body, throughout our bodies, and throughout all forms of life. In order to explore that more deeply, we must follow the flow of information in cells. And in cells, information is stored in the DNA. Now, DNA is a double-stranded helix of tightly wound genetic information that is itself wound around a set of proteins called histones that are themselves then tightly wound around each other into chromosomes. It's analogous to those old tape drives in IBM computers where in order to access that information, what cells have to do is they have to go find that right tape drive, unwind it, and start to make a working copy. That process of unwinding and making a working copy is called transcription, where a series of proteins will search that DNA, unwind it a bit, bind to the DNA, and start a process of making a pre-messenger RNA molecule. That pre-messenger RNA molecule, when done, will go off and be spliced by a whole different group of proteins called the spliceosome that will edit out certain pieces, leading to a messenger RNA. That messenger RNA is the first fully intact copy, working copy of the information of life that then leaves the nucleus and goes and does all of the work of life. It does that by seeking out ribosomes in the cytosol. These ribosomes translate the messenger RNA into all of the proteins of life, including secreted proteins, transmembrane proteins, mitochondrial proteins, and even proteins that go back into the nucleus and do all the work of storing that information or selecting information to be unwound to make more messenger RNA. So messenger RNA really is the central molecule in all of biology's processes. We call it the software of life. Now, given that power, it's reasonable to ask whether others have pursued it as a therapeutic approach. And in fact, many of the more exciting technology platforms in biotechnology today are. One example is siRNA. siRNA specifically binds to and degrades messenger RNAs inside of cells. It selects a specific messenger RNA relative to others, thereby changing the flow of information. Another example is gene therapy. In gene therapy, DNA is introduced from the outside and translocates into the nucleus of cells, where either episomal DNA or DNA that is inserted into the genome of the host will go through those same processes of transcription into pre-mRNAs and splicing into final mRNAs that then get exported out of the cell. In many senses, gene therapy is an indirect way to increase or add a specific type of messenger RNA to cells from the outside. So all of these approaches are about modifying the types or amounts of messenger RNA inside of cells so that you can have a phenotypic effect in a disease. Our approach has been more direct. Can we put messenger RNA directly into cells to create proteins, both endogenous proteins and foreign proteins? And if we could, we believe that messenger RNA could be used to make the full set of proteins in life that might allow us to treat an incredibly broad range of human diseases.
mRNA is literally messenger RNA. So as the word implies, message. It is a fundamental way to translate information from our genes, the DNA, to proteins. So every cell in our body makes different proteins. That's why our cells are different. But almost every cell in our body has the exact same DNA. So how do cells make different protein based on the same templates? And the answer is messenger RNA.
To understand what messenger RNA is, we first have to talk about proteins. Proteins are the building blocks for our body and how our body functions. When a protein or a set of proteins don't work properly, this can lead to serious health consequences. Messenger RNA, we think of it as a software of life. It's a set of instructions that we can provide to cells to tell them how to make specific proteins.
So at Moderna, we've been down this road now for a while. In fact, to date, we've treated over 1,000 healthy people and patients across a range of therapeutic applications. And it's not just us. Recognizing the potential of this platform, we partnered relatively early with leading pharmaceutical companies like AstraZeneca and Merck for some of our applications. And so not only are we conducting clinical trials for our drugs and vaccines, we actually have partners who are taking our drugs and then they're running the clinical development programs for those medicines.
So mRNA 3704 is an investigational product that has multiple designations from health authorities such as FDA and EMA, European Medicines Agency. mRNA 3704 is currently being evaluated in a phase 1/2 trial in patients with methylmalonic acidemia that have that methylmalonyl-CoA mutase deficiency. The trial is currently recruiting. We have open sites in the US and we hope to have open sites in the UK soon. So messenger RNA is a new type of medicine class that can be used to make really difficult types of proteins that traditional methods of medicine could not address previously. In support of the overall development program for mRNA 3704, we're also conducting a natural history study in which we want to characterize the signs and symptoms of patients with MMA and PA to help us understand these disorders better and potentially use these data to identify relevant trial endpoints that could be used and studied with investigational medicines. So mRNA 3704 is different from current options that focus on modifying the diet or focus on other supportive aspects. With mRNA 3704, we aim to address the underlying cause of the disorder by providing instructions to make that mute protein. To learn more about the phase 1/2 trial in which we're evaluating mRNA 3704, you can go to the clinicaltrials.gov listing. You can also go to the Moderna TX website. We're also working really closely with patient advocacy groups, specifically Organic Acidemia Association and Metabolic Support UK. And of course, you can contact your doctor to ask about this trial. We have a really talented group of individuals that work day in and day out to push hard in support of our mission to bring this new class of medicines to patients.
So CMV is a virus, it's a DNA virus. It's a virus that infects most people in the world. It's a virus that hides. Most of us don't know we've got it. And most of us go through life without it ever causing a problem.
If a woman during pregnancy is infected with CMV, they can transmit it to the child in the uterus, and that results in congenital CMV infection.
Every year there are about 20,000 children in the US born with CMV infection. Of them, about 6,000 to 7,000 will end up with deafness, with blindness, with brain damage, with microcephaly, with lifelong disabilities for many of them. Even if we were to look for it and measure it, there's really very little we can do about it at the moment.
And also, it's a severe threat in immunocompromised individuals, which includes HIV patients, transplant patients, both solid organ as well as in a stem cell transplant population.
The US Institute of Medicine made it very clear a number of years ago that the CMV is the #1 unmet vaccine need. So clearly everybody agrees this is a problem that needs to be solved. We're committed to do that, and we believe we have the tools to do that. As I said, there are 20,000 children born every year with this infection just in the US, and 6,000 to 7,000 of those every year will have severe disabilities. Just imagine if we can produce a vaccine to prevent that. CMV enters the cell really through two cell types. It enters through what are called fibroblasts and enters through what are called epithelial cells. The key to get into fibroblasts is a GB protein, and that's quite easy to make. The key to open up the epithelial cell is a pentameric protein. It's a protein made up of 5 different proteins that must assemble in exactly the right way.
We were simply able to design mRNAs encoding for the 5 different subunits of the pentamer and co-deliver them into a cell, and the cell's machinery just took over.
What we found and what this study shows is that if you insert the mRNA for those 5 proteins into the body processes them exactly as it would if it had been infected by the virus. And it expresses that protein beautifully, induces an immune response beautifully, and our expectation, and what we saw in animals, is that that immune response could protect against CMV infection. What this data tells us is not only that we have a really good shot at CMV, that it also means that this platform is likely to give us a good shot at any other complex vaccines. And not only single diseases that require multiple proteins and multiple mRNAs, but maybe even single vaccines to protect against multiple different diseases with a single injection.
It's intense.
Intense.
It is really intense.
Groundbreaking.
Diverse.
Meaningful.
Ambitious.
Achieve impossible with being impossible.
Disruptive.
A wonderful craziness.
It's really about making a difference.
It's amazing.
It's really thinking about the patients.
Like a way of life.
If you're willing to do something different, be creative, be a part of something big, this is it.